Polymerase chain reaction, PCR, in a technique used to amplify DNA samples. PCR enables the generation of sufficient DNA for analysis from even-minute samples. It involves separating DNA strands using high temperatures, which can denature most enzymes. The breakthrough came with the use of Taq polymerase from the bacterium Thermus aquaticus, which thrives in hot springs and can withstand the required temperatures. PCR is significant in forensic investigations, where small DNA samples can be amplified to create DNA profiles. The key steps of PCR are as follows. Denaturation. The mixture is heated to 90 to 95 degrees Celsius to separate the DNA strands by breaking the hydrogen bonds between them. Annealing. The mixture is cooled to 50 to 60 degrees Celsius to allow primers to bind anneal to the single DNA strands. Extension. The mixture is heated to 75 degrees Celsius, the optimum temperature for a Taq DNA polymerase, to build the complementary strands of DNA. These three steps are repeated around 30 times, amplifying the original DNA sample. 1. Describe how DNA can be amplified in the polymerase chain reaction. DNA amplification in PC involves a cyclical process with three main steps. Denaturation, the reaction mixture containing the DNA sample, Taq polymerase, primers, nucleotides, and buffer, is heated to 90 to 95 degrees Celsius. This high temperature breaks the hydrogen bonds between the DNA strands, separating them into single strands. Annealing. The temperature is lowered to 50 to 60 degrees Celsius, allowing the primers to bind anneal to their complementary sequences on the single-stranded DNA. Primers are short DNA sequences that provide a starting point for DNA synthesis. Extension. The temperature is raised to 75 degrees Celsius, the optimal temperature for a Taq DNA polymerase. This enzyme then adds nucleotides to the primers, extending the new DNA strand in the 5 feet to 3 feet direction, using the original DNA strand as a template. These three steps, denaturation, annealing, and extension, constitute one PCR cycle. This cycle is repeated around 30 times, with each cycle doubling the amount of DNA. This exponential amplification results in a large quantity of DNA from the initial small sample. 2. Explain why the use of enzymes from the bacterium Thermus aquaticus was such a breakthrough. The use of enzymes from Thermus aquaticus Taq polymerase was a breakthrough for PCR because of its heat stability. Previous attempts at PCR were hindered by the need for high temperatures to denature DNA, which also destroyed the DNA polymerase used for replication. This required the addition of fresh enzyme after each cycle, making the process inefficient and time-consuming. Taq polymerase, isolated from a bacterium that thrives in hot springs, is adapted to function at high temperatures. It remains active during the denaturation step and can withstand repeated heating and cooling cycles. This eliminated the need for a manual addition of enzyme in each cycle, automating the process and making it significantly more efficient and practical.